Briefly go over the 4 steps of cellular respiration. Remember that cellular respiration is the process that our cells is going to use to break down food and to transfer the energy in that food to ATP. And for every glucose molecule, we're going to make around 36 to 38 ATP. I love this illustration showing us food transferring its energy into ATP and then ATP transferring its energy for the cells to do work. Glycolysis is the first step of cellular respiration. This occurs in the cytoplasm of the cell. One glucose is broken down into 2 3-carbon molecules, and we have to use 2 ATP for this. I always tell my students you gotta spend money to make money. These molecules are converted into 2 3-carbon pyruvate molecules, and we produce 2 NADHs and 4 ATP. However, we're only producing 2 net ATP since we used 2 ATP in the beginning. The prep step is next. These pyruvate molecules are going to move into the matrix of the mitochondria. Pyruvate molecules are going to lose a carbon in the form of CO2. During this reaction, they are also going to produce an NADH, and then be converted into a molecule known as acetyl-CoA. Now, this step has to occur twice since we're getting 2 pyruvate molecules from the first step, glycolysis. The third step within cellular respiration is known as the Krebs cycle. This also occurs in the matrix of the mitochondria. Acetyl-CoA is going to enter the Krebs cycle where many enzymes are going to react with the molecules involved. And there are a lot of reactions that occur here, but focus on the products. For every acetyl-CoA, we produce 3 NADH, 1 FADH2, 2 CO2s, and 1 ATP. But remember, this cycle has to occur twice because we're getting 2 acetyl-CoAs from the prep step. The last step is going to be the electron transport chain. This is one of the more complex steps. The electron transport chain occurs on the inner mitochondrial membrane. All of those NADH and FADH2 molecules that we produced before are going to donate 2 things to the inner membrane. These molecules will donate electrons to the membrane proteins and hydrogen ions into the matrix. The electrons are going to move through the electron transport chain and pump hydrogen ions into the inner membrane space. At the end of the electron transport chain, these electrons will bind with an oxygen molecule along with hydrogen ions to form water. So oxygen is the final electron acceptor in this process. As more and more hydrogen ions move into the inner membrane space, you're going to create a concentration gradient. There will be a higher concentration of hydrogen ions in the inner membrane space compared with the matrix. And these hydrogen ions will move through an enzyme called ATP synthase. ATP synthase will act almost like a dam and use the movement of hydrogen ions to bind a phosphate group onto ADP. This makes ADP into ATP, which is a high energy molecule. And we make around 34 ATP per glucose at this step. Really quickly, you can see NADH and FADH2 donating their electrons and hydrogen ions here. These electrons will move through the electron transport chain and bind with oxygen to form water. We'll have a high concentration of hydrogen ions in the inner membrane space. These hydrogen ions will move through ATP synthase, which makes it spin and bind a phosphate group onto ADP, making it ATP. And this produces the most ATP during cellular respiration.